In this video, I'll be walking you through the procedure for genetically sexing Xenopus laevis. There are 3 steps to this procedure. First, DNA extraction, second, PCR, and finally, visualizing the results using gel electrophoresis. We'll first walk through the process for extracting DNA. So when removing the tissue, it's important to flash freeze it upon euthanization of the organism and hold it at -80 degrees Celsius until you're ready to analyze it. So start by removing 25 milligrams of tissue, avoiding bones and skin, as this can clog the filter of the spin column. It's also important to ethanol rinse your forceps and scalpel between animals. Once you have your tissue sample, place it into a 1.5 ml tube along with 180 microliters of ATL buffer and 20 microliters of proteinase K. Next, vortex the tube to mix it, ensuring that the tissue is at the bottom of the tube when you are done. Finally, place these tubes into the heat block set at 58 degrees Celsius for at least an hour. At the end, the tissue should be liquid. The PCR portion of this procedure is conducted following the QIAGEN DNA-EZ procedure. So once the tissue is liquid, you can remove it from the heat block and open all of the tubes and pipette in 200 microliters AL buffer and 200 microliters of 100% ethanol. Then transfer 600 microliters from your 1.5 microliter tubes into a spin column, discarding the old tube. Vortex the sample and then centrifuge for 1 minute at 8,000 RPM. Then add 500 microliters AW1 wash buffer, centrifuging again for 1 minute at 8,000 RPM. Move the filter portion from the spin column to a new collection tube and add 500 microliters of AW2 wash buffer and centrifuge again for 3 minutes at 14,000 RPM. Transfer the spin column filter to a new 1.5 ml tube and add 200 microliters of AE elution buffer. Centrifuge this for 1 minute at 8,000 RPM, and the final extracted DNA product will be at the bottom of the 1.5 ml tube. After extraction, you'll put the tubes into the thermocycler to amplify the DNA before analysis with the nanodrop spectrophotometer. The procedure for the spectrophotometer will be determined by the machine that you have, but essentially you will blank with the ATL buffer, and then when analyzing the results, your 260-280 ratio should be greater than 2, and your 260-230 ratio should be greater than 1.8 nanograms per microliter. Finally, the concentration should be greater than 50. If you find your numbers are outside of these ranges, you should not be using them for PCR and will need to go back and troubleshoot the procedure up to this point. If your DNA concentration was adequate, you can move on to the gel electrophoresis to visualize the results. So you'll make a 3% agarose gel using a ratio of 43 milliliters of 1% TAE to 1.2 grams of agarose, depending on the size of gel needed. Then add 10 microliters of CyberSafe per 100 milliliters of TAE buffer and fill the apparatus with 1% TAE buffer. Once you have your gel, you'll first load 5 microliters of a 25 base pair DNA ladder. Next, you'll load 2 microliters of the positive control into the second well, and finally 2 microliters of each sample PCR product into the subsequent wells. Run the gel at 160 volts for 30 to 40 minutes. You'll want to see the blue and yellow dye moving towards the red positive end. Once you've run the gel, you'll visualize it, and then you have completed your genetic sexing of Xenopus laevis.